Welcome friends. We continue with our discussion on mechanism of hormone action. In this video, we shall discuss G protein mediated signaling mechanism. The main components of the G protein signaling mechanism are the receptors, the heterotrimeric G proteins, and the effectors. This is the G protein coupled receptor. It is an integral membrane protein and has got 7 transmembrane segments 1, 2, 3, 4, 5, 6, 7. These are hydrophobic segments, and because of the presence of these 7 hydrophobic segments, the G protein coupled receptors are also known as 7 transmembrane receptors. There is another name for them, which is the serpentine receptors, because the segments are passing in and passing out of the cell membrane multiple times in a snake-like fashion. Each of the segment comprised of about 25 to 35 amino acids, and these amino acids, they form a stable alpha helices in the lipid bilayer of the target cell. The core of the G protein coupled receptor is common to all types of G protein coupled receptors, whereas the extracellular loops and the intracellular loops, they differ in the shape, they differ in the length, and they differ in the sequence of amino acids, which allows them to bind with different type of ligands and with different type of effectors inside the cell. The ligands bind to the extracellular loops and to the amino terminus of the G protein coupled receptor, whereas the G proteins, they bind the carboxy terminus of the G protein coupled receptor, and they also bind to the intracellular loops. This is the heterotrimeric G protein, so named because it is comprised of 3 subunits: alpha subunit, beta subunit, a gamma subunit. Alpha subunit binds either GDP or it binds GTP. Therefore, these proteins are named G proteins because they are G guanine nucleotide binding proteins. When the receptor is activated by the ligand, this receptor will undergo a conformational change, and once the receptor undergoes a conformational change, it is activated. The activated receptor will act as a guanine nucleotide exchange factor, and it will cause the alpha subunit to exchange GDP for GTP. Once GTP binds to the alpha subunit, this G alpha subunit dissociates from the beta gamma dimer. Then both the beta gamma dimer as well as the G alpha G protein subunit, they will independently act as signal transducers, and this G alpha subunit will cause the activation of intracellular effectors, including adenyl cyclase or phospholipase C. Activation of the adenyl cyclase and phospholipase C leads to the formation of second messengers. These include cyclic AMP, cyclic GMP, phosphoinositol triphosphate, and diacyl glycerol. The activated second messengers activate intracellular protein kinases such as protein kinase A, protein kinase C, which ultimately result in cellular response. Once the desired response has been achieved, the signal mechanism need to be terminated, and this is achieved by the inherent GTPase activity of the G alpha subunit. The G alpha subunit hydrolyzes its bound GTP and provides inactive G alpha GDP. The G alpha GDP reassociates with the beta gamma heterodimer and thus terminating the signaling of both G alpha GTP and the beta gamma subunits. Thus, the G proteins act as molecular switches inside the cells. GDP locks G proteins into their inactive state, while the GTP locks G proteins into their activated state. Besides, the magnitude and duration of cellular responses initiated by the G protein couple receptors can be modulated by regulatory proteins such as GTPase accelerating proteins. In short, these are known as GAPs. GTPase accelerating proteins increase the rate of hydrolysis of GTP to GTP. That means the hydrolysis of GTP to GTP will be faster. So the signaling will occur for a shorter duration of time. If they are not activated, in that case, the signaling gets prolonged. G proteins act as negative regulators of G protein signaling by serving as GTPase accelerating proteins or GAPs to switch off signaling by both the G alpha and beta gamma subunits of the heterotrimeric G protein. There are two signal transduction pathways involving the heterotrimeric G proteins: the cyclic AMP signal pathway and the phosphatidyl inositol signal pathway. The signal transduction depends on the type of G alpha protein which is activated by the ligand. There are different types of G alpha proteins like G alpha stimulatory, G alpha inhibitory, GQ proteins, and G transducin. Note the stimulatory G alpha stimulates the formation of adenyl cyclase, activates adenyl cyclase, and results in increased levels of cyclic AMP in the cell. The most important hormones which activate the stimulatory G proteins are adrenaline and parathyroid hormone. G alpha stimulatory is the target of cholera toxin. This cholera toxin, it is produced by the bacteria Vibrio cholera, and it can cause life-threatening diarrhea. Now the inhibitory G proteins, they inhibit adenyl cyclase and resulting in less formation of the cyclic AMP in the cell. An important hormone which activates the inhibitory G proteins is somatostatin. G alpha Q activates the effector molecule phospholipase C, and once activated, phospholipase C activates or generates the second messengers inositol triphosphate and diacyl glycerol. G alpha Q is found in G proteins coupled to receptors for ADH and thyroid stimulating hormones. G transducin is the molecule responsible for generating a signal in the rods of retina in response to light. G alpha T, that is transducin, triggers the breakdown of cyclic GMP.